But yeah, I came out of the high-tech market of computer, software, internet, and I was retired. I was, basically spending all my time on a beach in Hawaii. And believe it or not, after a while that gets boring. And I thought I'll just dabble in high-tech again. And then it was just luck that I ended up happening to get a business plan from NIH. And the guy who sent me the business plan introduced me to Roscoe Brady, who I did not realize when I met him. He was an incredibly accomplished, globally recognized drug developer, highly decorated scientist, and who was researching the most cutting edge of viral vector gene delivery. He sat down with me like a colleague or a peer, which is ridiculous. I mean, this guy was a giant in the industry. And he explained to me viral vectors. And coming from software, I was like, wait a minute. You're telling me that viruses are like diskettes now, where we can deliver genetic information that modifies DNA in human beings. I knew enough from my high school biology. I said, wait a minute. What can't we cure with this? We will send radiation and chemotherapy the way of bloodletting and leeches. The blind will see, the cripples will walk, the lepers will be healed. I mean, I can't believe what I'm seeing here. And he agreed with me from a scientific standpoint that these things were all possible. But coming from a high-tech background, I understand how these things are achieved. They're achieved because technology builds on technology, right? Drug development is nothing like that. Drug development is random discovery of things that are of value, and then you have to test and test and test and test and test, and it costs a fortune to do all those tests, and then usually you end up getting knocked out of your drug development. Why? Because there's a side effect, right? You didn't anticipate it. You made a new drug, you put it in the body, it goes to every cell. It's like the ultimate democratic thing that you take a pill and it makes no discrimination in your body. It basically delivers that drug to every part of your body. It treats every cell the same. And the problem is that it may be great on the disease in the cells that need it, but it has a side effect which makes it unusable. And this is a tremendous, it's the bane of drug development, right? It's really side effects that kills everything. Well, now with gene and cell therapy, even just remembering my 10th grade high school biology, now that we can, in a targeted manner, go in and tweak genes where we can target the virus itself to particular parts of the body, or we can use specific promoters to only turn it on in cells that are showing certain signs of diseases. We now can put genetic constructs, the medicine, exactly where we want it. Right? It'll only turn on where it's needed. We're sparing all these healthy cells from being treated by the medicine. Immediately, what that tells you is, wow, when you come up with something that's worth doing in a cell and you go ahead and implement it, you're going to be shielded from 90% of the stuff that keeps that drug from making it to the clinic. You can ratchet up the therapeutic effect. You can do something really powerful in the cells that need it. And you can spare that power, you can prevent that activity in the cells that don't need it. And so you've got a much clearer path to the clinic. But there's more to it than that. The ACTGs of your DNA are incredibly parallel to the zeros and ones of your computer. As a matter of fact, the way I see it, your human cells are nothing but organic computers. The instruction set is your DNA, and the DNA is in every single cell. Every single cell knows which part of the DNA to run. And you are trillions of collaborating organic computers where the order of those nucleotides, ACTG, makes up genes. Genes are commands to the cell. Your cell executes the part of your DNA that's appropriate for making light-sensitive protein in your eyes, making acid in your stomach, replacing skin cells that flake off, making your heart beat. and they communicate in this incredibly sophisticated way. I mean, look at yourself as like a corporation of cells, right? You got the brain cells that are, reasoning things out and sending instructions to the rest of the body. They're sending it electrically. Cells are communicating with chemicals. Cells are communicating even by bumping each other. So, this is what you have is an incredibly sophisticated computer, and it works in the materials that it's made of. The ACTG is just base 4. You could think of a gene as just being represented as a number. Same thing with zeros and ones. It's the order of those things that decide what the computer does. And now that we can go in and upgrade your DNA, upgrade your operating system, repair defects in your operating system, this is what I realized sitting there with Dr. Brady. The question isn't what can we do, it's what can't we do with that. Thousands and thousands of diseases are going to fall to this new powerful technology. I don't think anybody would be in this conference right now if they didn't already believe that. But the next question is, how can we get there as fast as possible? So how would you do it? If you realized, like I did that day, that reprogramming the human computer is the software industry of the next 100 years, how would you decide and how would you model your company to have the greatest impact as quickly as possible on the most diseases for the most people? You just look back at the last software industry. Would you want to be Lotus Corporation or would you want to be Microsoft? And what's the difference, right? Lotus Corporation was the #1 software company in the world in the early 80s. Your broker would have said, that's the company to buy. Nobody had even heard of Microsoft for the most part. But, well, that was not good. Isn't that sweet? That was my dad calling. So let me go ahead and silence my phone. He'll understand that. Look at Lotus Corporation. What did they have? They had the most incredible application software, right? The spreadsheet for the largest hardware platform. So they were the thing in the early 80s. But Microsoft had an advantage over Lotus. And that was they were more efficient software developers. What do I mean by that? They had a tool set that was reusable. It was essentially a whole bunch of components and technologies and know-how and intellectual property, and it was essentially subroutines. They'd done everything. They were experienced at so many different things, and they had so many different components that they could just reuse and leverage to develop new applications on top of that. So what are those components called? Well, in the software industry, it's called an operating system. In biotech, it's called a platform. And if you want to think about American Gene Technology's vision for the future of gene and cell therapy versus everybody else's vision, you just think of it as that we are focused on the operating system, where one day our operating system becomes like an iPhone. And if you want to cure a disease, you just write an app on top of that, right? All of the heavy lifting is already done. We have things that will reliably deliver the exact genetic construct to the exact right cells and the exact right therapeutic value or therapeutic levels safely, in a targeted way, highly specifically. This is something that needs to be done regardless of what you're curing in the human body, right? But then all you need to do is know the disease. What are the pathways in the cell that need to be severed to undercut the driver of that disease? What are the pathways that are missing that we need to enhance in order to mitigate a disease? Once you know that stuff, then you can work with that platform and you're 80% done on day one. Okay, that's the grand vision. So let me go in, let me just sort of dive into the market. I just want to talk to you about how big this is, because I think that everybody in this audience should be excited, regardless of where you sit in the value chain, whether you're a doctor, whether you are a researcher that wants to cure a disease, whether you are a CMO, whether you are CRO, whatever. This market is exploding. And, don't take my word for it. Trust Goldman Sachs, okay? Goldman Sachs predicts, and you can go ahead and look this up online yourself. There's a really neat video, and this is from last year, that this industry that we're going into together is $5 trillion. So let's compare that to the existing industry. The existing pharmaceutical industry is $1 trillion a year. 5x, according to Goldman Sachs, considered very credible when it comes to finance and markets and, and predicting the future of particular technologies. But I'll posit this to you. I think they're off by a factor of at least 2. I think there's a minimum of 10 trillion in this, because I can see where they come up with 5 trillion. It's simple. Monogenic loss of function disorders. If you're going to cure one of those, what do you need to do? You just need to find where in the body a broken gene or a missing gene is not making a gene product that you need to live. Not you, but the patients. Then you just need to deliver that gene by some viral vector and install it and turn it on in the right therapeutic level, in other words, in the right quantity. And guess what? You don't have a disease anymore, do you? Right? If I'm born with phenylketonuria because I don't have a working phenylalanine hydroxylase gene, well, if I can put that gene back in the cells that need it, then I don't have a disease anymore, do I? This is the idea of correcting the operating system where there's a missense copy somewhere, where you inherited a broken gene, right? We can now go in there and fix that by taking viruses, deadly viruses. We use lentivirus for the most part. We're agnostic to viral vectors. We just choose the viral vector based on the mission, okay? And I don't think of viral vectors as being a big deal. You're probably like, what? Well, I don't think of them as a big deal because I come from the computer industry, and what are viral vectors? They're just the physical layer for delivering the software. The software is the thing, right? What's the difference between AAV and lentivirus? Well, you could be really technical about it. Well, AAV is non-integrating, but it's really seldom integrating. So don't think of it as non-integrating. Think of it as seldom integrating. It integrates. And lentivirus is integrating. They have different attributes in terms of the cells that they transduce the best. A lot of AAV is targeted. You get the right AAV. AAV9 tends to go to the CNS. AAV5 tends to go somewhere else. So by picking your viral carrier, it's good for certain missions. But to me, the basic idea behind viral vectors is that they are the paper tape and punch cards of this software revolution. They are the earliest ways that we have to communicate with the computer, but they're not the last. All right? The diskette will come. Maybe that's CRISPR. The CD will come, the DVD will come, the thumb drive will come. And then eventually we'll be delivering these solutions over the internet. I know that sounds crazy, but I don't see a reason why I can't send you software to fix your disease if you had the right machine in your house to just produce the drug in your house, right? And I don't see that as being impossible. So software for this computer may move over the internet the way that software for your digital computer moves over the internet. So the future's endless, but the viral vectors are just about a physical layer for transferring the software. The software is the thing, and the application is the cure. And it's, sort of where we are in the value stack, operating system, but nobody would buy our operating system until we prove that it works. And we're going to prove that it works, I think, this year or early next year by curing HIV. So gene and cell therapy, just to sum it up, power to cure. It's got power to cure because it has the ability to go in and actually change the genetic drivers of disease. And since lentiviral vectors and other integrating vectors can put a permanent change in there, we can do one and done treatments that are actually cures. And power is really something to think about here. You can ratchet up the efficacy of drugs. You can go in there with a sledgehammer if you know that the sledgehammer's only going to hit what you need it to hit, right? And that comes down to specificity, or what I call smart drugs, smart bombs. The specific promoter is like a smart fuse on the drug, right? In other words, it only turns on where you need it. What can you use a specific promoter for? I could have a specific promoter that only turns on the drug construct when it sees high levels of human prostate-specific antigen. What might that be good for? How about prostate cancer? High levels of HPSA indicate prostate cancer. Kaboom! I could just slip that cell a apoptosis gene, and the cell will shut down and die. Or I could sever a pathway that might not be deadly to the cell, but might prevent it from rapidly dividing, making it your other therapies more effective. I mean, I can decide what to do, though, based on that smart fuse. That's the critical thing. What else is a smart fuse good for? Well, what if I only want this thing to fire in a certain type of tissue? Like, let's say I'm going to treat your liver because you have a liver-centric metabolic genetic loss of function disorder. An example of that is phenylketonuria, right? You don't have phenylalanine hydroxylase in your liver breaking down phenylalanine. Deadly buildup of phenylalanine is giving you pain every day, and you're robbed of the downstream byproducts of breaking down phenylalanine, which, by the way, is the most serious thing in phenylketonuria. You're tyrosine starved your whole life, and other nutrients that come from that pathway that assure nerve health in your brain. So if you're born with phenylketonuria, which, by the way, in the United States is about 1 in 13,000 babies. In Turkey, it's 1 in 2,500 babies. And you detect it by a blood test when the baby's born. In the United States, it's typical that we take 1 drop of blood from the heel of the baby and we test it for a lot of different things. But one of the things we look for is phenylalanine hydroxylase. And if we can't find it, that baby can never have animal protein. That's less of an issue, you know, for people that are vegetarians or whatever. But in the United States, where, you know, those types of foods are available everywhere, this is really serious. Right? You have to get all your protein from some other method, and you got to somehow stay away from all those foods that everybody else is eating. Otherwise, you'll get nerve damage and organ damage from deadly buildup of phenylalanine that can't be broken down. But regardless of how good you are about your diet, you're still going to be depressed by age 20, schizophrenic by age 30, and having suicidal thoughts by age 40. And it is only one broken gene in your body. And, okay, so let's say I want to treat that in the liver. So I'm going to give you a dose of something you need in the liver, but you don't need anywhere else. And in order to insulate you from side effects that could happen in your kidney or in your heart, if some of it gets out of the liver, how do I prevent that? I put a smart fuse on it that says, only fire this drug if you have high levels of albumin. Albumin is characteristic of the liver. Okay, so this is all about targeting that drug. And it's all part of the idea of specificity, avoiding side effects. You know exactly what you're doing to the cell. You're not putting a chemical that operates on the outside of the cell. You know, this could be very nonspecific. It could trigger a surface receptor that could have all sorts of consequences in the cell. No, you are going in and you are tweaking one pathway. You are upregulating a gene or downregulating a gene. You have incredibly specific effects that are predictable. This is the beauty of gene and cell therapy. This is what is leading to an unbelievable acceleration in the idea of curing diseases and, in fact, in achieving those cures. And I probably don't need to tell you, but there's like 7 or 8 already approved miracle cures in gene and cell therapy. How about this one? You're born with Leber's congenital amaurosis, you go blind, you get a drug that was originally developed by Spark that's now owned by Novartis. It's a few drops of an AAV vector for your eyes. It restores the protein that you are missing because you have a broken gene in your eye that leads to that blindness. and blind people are seeing. This is what got me so excited the day that I met Roscoe Brady, as I understood there are tons of diseases like that, and the reality is there's actually 10,000 monogenic loss of, 7,000 known monogenic loss of function disorders. All right. But some of these are very, very rare conditions. And how do we get these very expensive, you might be thinking expensive gene and cell therapies out to the people that need them? Well, if I have time, I'll talk about that later. But this is a market that, again, is very similar to the software and computer market that I come from. And that is that the capabilities double every year and the cost halves. So what does that mean? You may look from various locations around the world and go, yeah, that's great for the US and Europe, but how are we going to afford it in any place, besides wealthy, well-insured markets? Well, let me ask you this question. When IBM dominated the computing market, would you have believed that almost everybody in Africa would have one of these in their pocket? 10 times the power of the most the biggest IBM computer in the 60s that talks to you, that communicates with the world? No, that's what technology does. And the key element of technology is that reusable components build on one another. So it's not just that things are getting better and cheaper, it's that little things are being invented all along that sort of unlock capabilities for this general technical area. A lot of the biggest problems with curing monogenic diseases come down to finding good ways to mitigate the immune reaction when you put a missing protein back in the body, because if you've grown up all your life without that protein, your immune system will recognize that as an invader and try to attack it. This is one of the things that's holding up a lot of those things right now. But we're going to find a way to fix that. And that's going to fix it for many different diseases. So it's not only on this curve where it's just getting better and better and better and cheaper and cheaper and cheaper, and we'll drive anything that comes out will eventually be available in sub-Saharan Africa. It's that we're also inventing new components which mix in with the rest of the components and have this geometric effect on the capabilities. Let's go back to IBM in the 60s. Did you imagine that computers would drive your cars and fly your planes? Did you imagine they would talk to you and they would understand your voice and parse it and respond to it? Did you think that you'd have all the knowledge of the world in your pocket from the internet? Of course not. But people that understood the trends in software and computers could have predicted that. What starts as science fiction ends up as science fact. Science fiction is just vision into the future. I'm telling you, my vision is this is just like computers and software. If you're in this environment where it's a technology like computers, if you can dream it, you can do it. It's just a matter of time. So we will cure all the diseases, all 7,000 monogenic diseases. Now, going back to the Goldman Sachs report, guess how much the world spends on palliative treatments for monogenic loss of function disorders just to keep people comfortable until they die. Okay, remember, we're not curing them with all that care. All we're doing is mitigating some of the symptoms to try to make these patients more comfortable. $2 to $4 trillion worth of resources in the world. Imagine applying that $2 to $4 trillion into curing those people instead. You'd give them longer lives with higher quality of life. I mean, for the patient, it's just unbelievable. But in fact, we can cure things more cheaply than we can treat them with gene and cell therapy. And how cheaply we can do it is actually getting better and better from year to year. I know this because I've been in this business for 12 years, developing those reusable components. We have a $2.4 billion platform of components that can be mixed and matched to cure a myriad of diseases. So why haven't we cured everything? Well, first of all, we have limited resources for the clinical development. So what we have done is focused on 3 different products right now that we think will prove out the platform It'll also bring us enough money that we can broaden out our pipeline. So we're doing tens of drugs instead of, 3 drugs, or maybe even hundreds of drugs. It also means that we can take our platform, right? Once we've proven that it works, there are some people sitting right in this audience right now that'll realize that we can be a key to curing a disease that they are interested in. Whether you're a physician or a researcher and you understand the pathology of a particular disease, ultimately, when we show that our tools get you 80% of the way there the day you start, and that we can reduce your cost of early experiments that allow you to decide whether that drug will work, we've just opened up an opportunity for you to do something that you're passionate about. We're passionate about this whole vision and about this mission of bringing this efficiency to this new drug development modality and curing a bunch of diseases. But there are passionate people throughout the world that can use some of the things that we've done to do amazing things. And all we have to do is contribute our tool set. Okay, so what are we going to do? We're going to cure HIV. We think we're going to go into the clinic this year. I think you're going to see results from this in the middle of next year. So within 12 months, I hope you'll be reading across the globe that HIV can be reliably cured and that cure came from American Gene Technologies. I'll dive into that in just a moment. Then we have phenylketonuria. Sadly, phenylketonuria is like the poor stepchild between the other, sort of fair-haired children, because think about phenylketonuria as a relatively small market, and so HIV is working out so well that it's getting all the money. Meanwhile, we have another one on the other side. We have a cure for epithelial solid tumors. Imagine if you went to the doctor and you, if you were a woman, let's say, and they examined you for breast cancer and they said, oh, we found a lump and it is malignant. It is potentially metastatic. It is serious, right? Or let's say you were a man and you went into your doctor and they checked your prostate and they said the same thing, right? Those are both what are called epithelial solid tumors. It's breast, prostate, lung, liver, colon, kidney, ovarian, pancreatic, head and neck, skin cancers, and others. All right, so there's a lot of them in there. And they're very, very deadly. They grow in the epithelial layers, which are already rapidly dividing cells, and then they're cancers that just go nuts, right? Okay, so you're going to need treatment. What's the doctor going to say to you next? Surgery, radiation, and chemotherapy. Typical treatment for any of these advanced stage cancers. But what if instead we could just take a small amount of viral vector and inject it directly into the primary tumor? It would be a virus that would carry a transgene into only 20% of the cancer cells. That's all we need to hit. And those cells would start creating a stimulatory small molecule that would bind to the microenvironment of that tumor and create a site of immune response, a site of immunogenicity that would attract every type 2 gamma delta T cell in your body to that site. It would cause them to activate. When they activate, they eat at 300 to 600 times their normal rate. It would cause them to proliferate, and they would circulate all over your body. Well, I'll tell you what happens. We have a 1,000-mouse study that shows this. That primary tumor, obliterated, gone. But in the time that they're circulating around, it takes a certain amount of time to chew away that primary tumor, the biggest tumor. It's circulating all over your body. It finds the secondary tumors, the metastases, and even unrelated epithelial solid tumors. I kid you not. You could be treated for lung cancer, and if you had some breast cancer that they hadn't detected or some prostate cancer they hadn't detected, it would mop that up at the same time, because the gamma delta T cells don't know the difference between any of those cancers. What they exquisitely, specifically know the difference between is malignant cells and non-malignant cells. They have never been seen in science, research, or medicine as ever attacking the wrong cell. There are diseases in which these gamma delta T cells amplify up to 95% of your circulating T cells. So this is safe. In those diseases, you don't die. You might feel a little bit achy from that kind of immune response, but they don't create cytokine storms like CAR T. They kill with granules instead, so you don't get the same reaction. But that's the story, is that they have an NK receptor that will only trigger if it senses the very complex set of stress molecules that are characteristic of human epithelial malignancy. And when that thing triggers, it becomes cytotoxic and kills that cell. And when that thing is activated by this immune, it's almost like a vaccine, but it's not a vaccine. I caution you, this is not a vaccine against cancer. But it has that sort of effect in which it's training your immune system and activating your immune system to rise up in strength and levels to clear a pathogen. In this case, the pathogen is cancer. And it gets it from everywhere in your body by just treating one tumor, the biggest tumor, the primary tumor. So think about your diagnosis now, right? Breast cancer. Surgery, radiation, and chemotherapy? No. You're going to need a shot. They can take a syringe and put it into the primary tumor and squeeze out the right amount of vector. And what are they going to tell you? Come back in 6 weeks so we can image you and make sure we got it all. End of treatment. This was my dream. Now, we're not as far along on gamma delta T cells as we are on HIV. I mean, HIV, we literally have a study from the National Institute of Allergies and Infectious Diseases. Has anybody heard of Tony Fauci recently? He's famous in HIV. He's become more famous for COVID. But the reality is that that is one of the top labs in the world. And when we showed them our preclinical data, we made a cell product that is so potent at clearing HIV that it is orders of magnitude better than anything that has ever been seen before. That includes antiretroviral therapy. So we can take these cells, put them in your body, and they take over for the antiretroviral therapy and suppress the virus so well that patients without antiretroviral therapy should be able to. Now, I'm saying should because we have to put it back into the patients to prove this. But based on our in vitro experiments, our ex vivo experiments on this cell product and challenging it with HIV and latently infected cells and seeing its ability to clear things, that anybody who gets these cells can throw away their antiretroviral meds. They can never get AIDS, they can never pass this virus to anybody else, and they can never be reinfected. And so we showed that preclinical data, that in vitro data from our cell product that was made from the blood of HIV-infected individuals who were volunteers in a clinical trial to give us their blood. And we have that blood product, and we showed that to NIAID, and they said, we wouldn't even believe this data unless we reproduce this data in our own lab. So we gave them all our reagents, they bled their own patients, and they have just written an article that's published in Molecular Therapy that documents the potency of this. So this has been validated by a really great third party, and that's why I'm so confident about HIV. Okay, so gamma delta T cells, well, our collaborator there is the Stanford Medical School. You may have heard of them. And a professor there and clinician by the name of Dr. Dean Felsher. He is a globally recognized expert in hepatocellular carcinoma, liver cancer, the most deadly form of liver cancer. He's been a collaborator with us for 5 years. We think we can get this product into his clinic in the next 24 months. And we think he'll put it into liver cancer patients, and we will see the same sort of remission that we're seeing in our mouse studies. In our mouse studies, we're getting 85% of the mice come back completely cancer-free, zero cancer, by just treating the primary tumor. And this is in advanced stage models of human cancer in a mouse using human gamma delta T cells that we put into the mouse and they do nothing. Then we go ahead and treat a tumor, and it eliminates that tumor plus any other tumors that we put on the body. This works even in mixed models. I could put liver cancer on one flank, breast cancer on the other flank, and treat the liver cancer. The breast cancer goes away as well. Just need to get the largest tumor to get this effect. Anyway, it's tremendous. So PKU is getting a little less attention in the middle there, but that's something that's getting a lot of interest from pharma companies now. So I think we may out-license that. That'll give it plenty of money to go into the clinic. And I think we can get into the clinic on PKU by the end of next year with enough money. All right, let's talk HIV. I'm going to give you the specifics of how we're curing HIV because I think that it'll help you to understand why I'm so confident. You've heard about the data already, but let me show you the process by which it's done. It's called an autologous cell therapy. Okay, autologous, we're taking cells out of somebody, modifying them ex vivo, and putting them back in. What does that sound like? Sounds like CAR T, right? Okay, it is. It's very similar to CAR T. We are filtering the blood for immune cells, and then we are isolating the immune cells that we care about, and we're modifying them, and then we're reinfusing that back into patients. The cell product, the modification, allows HIV T cells to really work against HIV. Because why do you get HIV? Comes into your body in a similar fashion to a cold or the flu or whatever. I mean, it's not airborne, thank goodness. But a viron somehow gets into your body, and you have T cells for HIV, but why don't they work? They actually run right over to the first infection. So the HIV T cells will be the first T cells to arrive at the pathogen. And the problem is that in HIV, the pathogen has evolved the capability to grab onto that T cell and infect it. The T cell that should have been your protection, the sentinel T cell that was there to detect the pathogen and start the entire immune cascade against the pathogen, is now the beachhead for the infection. It's as simple as that. And it's been proven in experiment after experiment after experiment, and there are certain people even in Northern Europe that are immune to certain strains of HIV, the most common strains that are called R5 strains, because they're missing one receptor on that T cell, which makes it hard for the viron to bind to the T cell, which makes it hard for the viron to infect the T cell, and guess what happens? The T cell does its job and clears the pathogen. Now, it's some small percentage of people that are born immune in Northern Europe. But they've proven that if you take bone marrow transplants from those people that are immune and you put them into HIV positive patients, those patients become immune. Why? Because they are missing that CCR5 receptor that the viron's looking for, the same way as the person that was naturally born. But you wouldn't want to give bone marrow transplants to every HIV patient because, first of all, 6 of 10 people that get a bone marrow transplant die from the trauma of the bone marrow transplant. That's how serious it is. But if you could give that capability to an HIV positive person without having to do a bone marrow transplant, and if you could do it cheaply and safely, that's a cure for HIV. That's a known factor. Okay, so what did we do? What we designed is this. First stop, leukapheresis. It's a blood draw basically, but it filters for T cells. So what we end up with is 400 milliliter leukopak. So a little bit smaller than a blood donation, but it is filtered for T cells, so it's mostly white blood cells or a high level of blood cells in there. It's not dangerous to take a leukapheresis like that. Your bone marrow will refill those T cells right away. This is even true in a person that has HIV. If you are well controlled on antiretroviral therapy, you now have full diversity of your T cells, including HIV T cells. So when we take that leukopak from an HIV-infected individual who's well controlled, we have what we need in there, HIV T cells that we want to boost in a way where they can actually protect this person against HIV once we put them back in. Okay, next stop is a machine. Why a machine? Look, this has got to be, if we're going to commercialize this, it's got to be distributable, right, and scalable. So what we did was we found a $250,000 machine that could automate this whole process. Put the blood in one side, push a button, and on the other side you get the blood product. And then that blood product needs some testing before you need to make sure that it all worked correctly. But in all the experiments that we've done, and we've now done this in over a dozen patients, so we've made over a dozen blood products, we're getting better than 9 out of 10 of them work. So we get a blood product in this automated cell protocol. All right? So this machine, any hospital could buy it. It's $250,000. It doesn't need a clean room. It needs an operator that's trained and then they could be going ahead, if they can do a leukapheresis and a reinfusion, they could be treating HIV. That's the idea. Okay, so you're in the automated machine. What does it do? Well, over the next 11 days, it will first stimulate the T cells that are in that leukapheresis. And we do that with a special set, proprietary set of stimulatory peptides that stimulate different portions of the GAG protein. And what that does is it tends to make the HIV-specific T cells proliferate in the mix. Now we have a higher level of HIV T cells than we had originally. And then the next thing that we do is we do a depletion protocol where we get all the HIV T cells to stay in the mix, and we throw away the ones that are non-HIV. Now, why would we do that? Your cold T cell only sees an HIV viron because the HIV T cells are not doing their job of clearing them. So if your HIV T cells are in sufficient quantity, and if they can't be infected and depleted by HIV, they will clear the HIV viremia to the extent that none of the other T cells matter anymore. They'll never see a viron. And if a viron infects another T cell, an HIV T cell will attack that T cell and destroy it, get rid of it. So the point is what you need is HIV protection. You don't need every cell in your body protected against HIV. So why do we want to only do the HIV T cells? Because also to commercialize this, it has to be as inexpensive as possible. Previous experiments that have tried to do things like this have used methods that can cost millions of dollars to treat a single patient. They use, the last experiment was Sangamo. They used $500,000 of viral vector for the treatment. Once we get the proper T cells isolated and we treat them with our viral vector, we're only using $35,000 worth of lentiviral vector for this whole process. That's one of the biggest costs associated with this whole thing. So we're doing it for less than 1/10 of the cost of the last attempt on this. By the way, that last attempt only worked in 1 in 10 patients, and they had to be what's called the heterozygous delta 32 mutant. They had to be missing half of their CCR5 anyway in order to do it. So, they were using $500,000 worth of vector, and they were getting 1 in 10 patients being cured. Think about, that's just impractical. It's not going to work in terms of commercialization. But it does prove our point, which is then we have HIV T cells which are uninfectable by HIV. If they're uninfectable by HIV, what do? They kill HIV. That's the whole idea. If you can't deplete them, if you can't destroy them, if you can't infect them and make them part of the infection, then what will happen is they will do their job and clear HIV and protect you. Then we want to say, how many do you need? How many of these supercells, HIV T cells that cannot be killed by HIV, do you need in your body to clear HIV? So you ask immunologists, what's the normal amount of these CD4 positive T cells, the helper T cells that start the entire immune cascade? What's the number that you normally have after you clear a pathogen, like let's say a flu or a cold or something like that? And they'll pretty consistently tell you about 100 million of them. Well, what we do is we culture up our HIV T cells to 1 billion, 10 times that number. And we cryopreserve them. We send them out for a set of release criteria because we need to make sure that everything worked properly. That's the nice thing about ex vivo cell manipulation is you can test them and make sure they're good before you put them back in the patient. But after that, you just reinfuse it into the patient. 1 billion cells should be somewhere between 3 and 10 times more than they need to keep them immune to HIV for the rest of their life. They can throw away their meds, they'll never get AIDS, they'll never be able to infect anybody else, and they can never be reinfected. And by the way, this is better than a bone marrow transplant in some ways, besides the fact that this process is unlikely to kill you. All right, there's some possibility it won't make you better. And we'll prove it in these upcoming human trials that it'll make you better. I think we're going to see a high level of efficacy, but at least it's a relatively non-traumatic process. CAR T has already proven that more cells than this with a more toxic modification can go into your body and it doesn't kill you. All right, we're not even changing the function of those T cells. CAR T changes the function of T cells. That's somewhat dangerous. It works. I'm not denigrating CAR Ts. But what we're doing doesn't change the function of those T cells at all. They still only have one antigen receptor. They're still only looking for one pathogen. All we're doing is making it harder or actually impossible for HIV to get into them. And we're protecting them not just from the infiltration of R5 strains, because R5 is the most common strain on Earth, and that's the one, if you're born without CCR5, you're immune to R5 strains. But there are X4 tropic strains. They don't need the R5, the CCR5 to get in. Well, what we do is we actually install a gene in all these cells that creates an siRNA against conserved regions of all genomes of HIV, all known strains, all known clades. What does that mean? It means that if something gets past the membrane, then there'll be siRNAs that will clip the DNA before it can integrate. Or if you even have a pre-infected cell, it will shut down the production of HIV virons. It sort of returns the cell to normalcy because we have siRNAs against genes that are required for creating HIV virons. We actually go after VIF and TAT. Okay, so that's all very technical, and I think most of you probably understand that. But at the end of this thing, we think we have a cure for HIV. And so where are we in the process? Well, what we've done is we have submitted an investigational new drug in the United States to the FDA. We did that in October of 2019, so that's a while ago. They're overwhelmed with INDs. It took them a while to really, be able to get the right experts there on this particular IND, because you have to be an expert in virology, immunology, and HIV to evaluate this. This is cutting edge stuff, obviously, and they're very concerned about safety. But it looks like they kind of get it at this point, and I think that in the next couple of months, there's a good chance that they'll feel comfortable and they'll allow us to start the human trial. We already have 3 hospitals signed up. We've got volunteers literally coming over the transom, so we won't have any trouble getting volunteers on this. So I think we're going to treat our first patients this year, and I think we're going to have data early next year, and we're going to do measurements of the blood that will prove to scientists that this therapy is working and that it would be safe for these people to go off of their antiretroviral therapy and that they should be immune for life. The reason they're immune for life is that the low amount of stimulation that you get from the viral reservoir will keep these cells active in the body as long as there is any viral reservoir. Once the viral reservoir goes away, what happens is that these CD4 positive helper T cells will retreat into the bone marrow, and they can reemerge very quickly if you're reinfected. So that's why we think this is lifetime protection. Now, as I was mentioning, we showed all that preclinical data because we've run that process on over a dozen patients. And we showed that preclinical data to NIAID, the National Institutes of Allergy and Infectious Diseases, and they were excited. They took our reagents. We gave them our viral vector. We taught them our process. They went ahead and executed it. They bled their own patients. They made the cell product. They did the HIV challenge tests. They got the same data. It's now documented in Molecular Therapy in an article that was co-authored by them. So that's a really exciting third-party sort of validation of what we're doing. It makes me even more confident that we can cure this. I'll tell you one other thing just real quickly that makes me very confident that we've got a high likelihood of providing a functional cure. Remember I mentioned Sangamo? So Sangamo did a study where they use what's called zinc finger nuclease clipping of the CCR5 gene on bulk T cells removed from patients. Okay, so they take an HIV-infected individual, they removed T cells, they used $500,000 worth of vector to go in there and clip all the CCR5 they could. Now remember, you can only drive that reaction so high before you get off-target clips. So they ran it as high as they can. And what happened was in the first study, they had 1 patient out of 12 in remission. They were basically cured of HIV, and that was a very exciting result. But the issue was 1 in 12. Why 1 in 12? Well, it was actually our lead, well, our chief scientific advisor knew Carl June, who was running that study. Our chief scientific advisor, by the way, was a guy named Bob Redfield, who now runs the CDC. So you've probably heard his name as well. Anyway, he picked up the phone to Carl and he said, you ought to test that one person that it cured to see if they were a heterozygous delta 32 mutant. In other words, problem with zinc finger nuclease clipping is you have to get both alleles in every cell. And if you only get one allele, there's plenty of CCR5 still left over for that cell to be infected by HIV. Well, it turned out that Bob was right. That one person was a heterozygous delta 32 mutant. They already had one broken CCR5 gene, and Sangamo had managed to break enough of the other allele that they got a therapeutic benefit. So then Sangamo reran the study on only people that had one broken gene. They still only got 1 in 10 patients. All right, well, that's a little discouraging in a way because that sounds like a good experiment, but here's the key. If you measure the number of HIV T cells that were returned to those patients of Sangamo that went into durable remission, that were depleted of CCR5, you can determine what that number is in their whole body, right? Remember, the HIV T cells are the only important ones. So you calculate that number and you compare it to the number of T cells that are immune, that are HIV specific and immune to HIV that we return, 2,000 to 1. Get that? They were getting 1 in 10 patients. That's why I'm fairly confident that we're going to do better than that. But only the human trial will prove that. But I'm hoping that we're going to start that human trial later this year. Okay, enough on HIV. Let's talk about immuno-oncology real quickly, and then I'll just open it up for questions. I hope there's still some people out there listening to me and that you have some questions. So maybe send them to the moderator or whatever, so she's getting ready. But I should be able to wrap this up in about 5 minutes or so, 5 to 10 minutes. So immuno-oncology, how do we do this? So why do you get a solid tumor? I mean, you have malignancy all over your body right now. Why does some of it turn into a solid tumor that becomes a threat to your life and some of it not? We all probably have different opinions on why that is. I'll tell you my theory. My theory is this. You have a system for eliminating small malignancies, and it's called gamma delta T cells. Type 2 gamma delta T cells. They spend their entire existence just looking for areas of rapidly dividing cells in your body, and they test all those cells with an NK receptor, and if the NK receptor triggers, they kill that cell because they know it's malignant. All right, and 3 to 5% of your circulating T cells are that type of T cell, which is remarkable, right? Think of all the diseases that you are prepared for in your body. Thousands and thousands of diseases that have some T cells in your body, right? These CD4 positive T cells are ready to detect the pathogen for, hundreds of different types of colds, hundreds of different types of flus, hundreds of types of bacteria, hundreds of types of fungus. They have antigen receptors that are sensitive to certain pathogens, right? So the diversity of your T cell subset is amazing. You even have T cells that are for diseases that haven't been on the planet in thousands of years. And most of the T cells that you're born with will die with you, never having seen the pathogen that they were ready to protect you against. But still, what is there? That's 3 to 5 out of 100 T cells, 3 to 5% are these gamma delta T cells. They are in memory levels and they are active all the time. And all they're doing is testing cells that are rapidly dividing to make sure they're not malignant. And if they're malignant, they are eliminating them. And it turns out that 2 out of 3 people will go their whole life without getting an epithelial tumor, something that needs to be treated in the clinic. That's remarkable. So these things do a great job. But here's what I think happens. As you age, the accrued stress on your cells is increasing, right? That makes sense. And the immune system over time, we know, can diminish a little bit. So you'll hit some critical mass point where these small malignancies may actually end up, sort of being near each other, and maybe they, 3 cells manage to turn into 6 cells, turns into 12 cells, and somehow it's blowing up like a balloon. And just out of bad luck, the gamma delta T cells didn't show up on time to eliminate it when it was 1 or 2 cells, and now it's ahead of the gamma delta T cells' ability to eat these things. So that's how you get a solid tumor. So now you have a solid tumor. And solid tumors are really tricky things because, as you know also from other types of immuno-oncology drugs like Keytruda or checkpoint inhibitors, they can actually throw things up. They've evolved to throw things up on their surface so the tumor starts to look like an organ to your immune system. In other words, they have these camouflage, these checkpoint molecules that try to fool your T cells into thinking this is just a normal part of your body, so it doesn't get attacked. So that's why you need surgery, radiation, and chemotherapy, because this thing could metastasize and go all over your body, right? All right. So remember I told you we could just do one shot to the primary tumor. That's exactly what we do. We take a little bit of this vector that we call immunotox, but basically it is a lentiviral vector carrying a transgene that severs part of the pathway for cholesterol. Cholesterol is in very high levels when cells are rapidly dividing, and it tends to throw off something called IPP that hangs around nearby the rapid division, and that's what attracts these gamma delta T cells. And it stimulates their T cell receptor, the gamma delta chain, and that activates them. Now, it doesn't target cells that have IPP. IPP is just a perfume. It's just neon lighting that attracts these things in. They only kill if the NK receptor gets triggered. So when we make this injection into the tumor, we're not going to get all the tumor cells, but we only need to get about 20% of them to get enough IPP binding to the microenvironment of that tumor to attract every gamma delta T cell in your body, activate it, proliferate them, and they start to eat at 300 to 600 times their normal rate. What does that do? Well, it does exactly what you'd expect. So in the activated state here, they're just going to turn on that tumor and they're going to obliterate it. Boom. All the cells will be dead. Not just the ones that are producing IPP, because that doesn't matter. All of the cells in the tumor are going to be detected by those NK receptors because this activation causes these cells to query every cell in the area, and when they find a match, kill it. So they don't just kill the cells that are producing IPP, they kill all the cells in the tumor. No more tumor. Okay, that would be good enough, wouldn't it? But there's a side effect, and it's a good one. These cells are circulating all over your body. What else do they see in your blood and your lymph? Metastases, secondary tumors. And they eliminate those with no further treatment. Like I said, in the mouse models, we put tumors all over the body of the mouse. We treat one, all the tumors go away. We frozen section the mouse, nothing left in those tumors besides gamma delta T cells looking for more malignant cells to kill and not being able to find any more because they're all gone. This is really, really exciting. 1,000 mice is a great preclinical result. This is a great model for humans. It should translate beautifully. We're essentially using the mouse like a furry little test tube, right? We're putting human cancers in its body. Then we're taking human gamma delta T cells and putting them into the peritoneum, nowhere near the tumors. And then we're activating those gamma delta T cells with our shot. Without the shot, nothing. Without the gamma delta T cells, nothing. But when you put them both in there, these things exquisitely, specifically hunt down and kill all malignancy in the mouse body. There's no reason to believe that this won't work the same way in humans, but we hope to prove that out in the next couple of years with our collaborator at the Stanford Medical School. So I hope the way you feel is the way that I feel, and that is gene and cell therapy has the ability to bring on miracle cures. Our methodology for developing drugs is to create a platform on which we can make many drugs more quickly, that are more predictable in the clinic, more reliable, and that we can address more diseases. And our platform is capable of doing that. And when you hear that we cured HIV, and I really believe you will hear that in the next year or so, I hope you'll read it right in your local paper. That will not just say that HIV may be history. What it will say is that the AGT model for efficiently developing new applications that cure diseases works. This is not science fiction, it is science fact. Luxturna proves that blindness, certain types of blindness can be cured. 2 CAR-Ts are curing incurable cancers. There are cures now for ACA SCID and X-SCID. Babies that are born without an immune system that had literally no chance of a life are now being cured and coming out of their bubbles and having normal lives. Spinal muscular atrophy, cured by Avexis, now owned by Novartis. Spinal muscular atrophy, when kids were born of that, they never developed their spine or the muscles around the spine enough that they could hold their head up on their own, and they die within, several years because they can't even breathe on their own. And now they can replace that one gene, and there's a cure for that. It's not 100% effective, and it's, relatively expensive, but it's cheaper than the kid dying. So all of these gene therapies make sense, and they're going to get cheaper over time too. They're going to get better, they're going to get cheaper. I think the Avexis cure, this is one of the things to watch for. There's multiple ways to do things in software. Just because VisiCalc wrote the spreadsheet didn't mean that it dominated the spreadsheet market forever. No, Lotus made a better one. And then Lotus didn't dominate even for 19 years, right? Because Excel, Microsoft made a better one. And that's the way this industry is going to work too. So if it costs us $2.1 million to cure somebody of spinal muscular atrophy today, I think you can assume that, wait a few years, it'll cost $500,000. Wait 10 years after that, it'll cost $50,000. I don't know how the drug market's going to price all these things. That's going to be a capitalist thing. That's not, a scientific thing. That's about profit maximization and corporations and government policy and all those things. But the bottom line is that we can make these things cheap enough. And if we can do enough drugs, we don't have to make maximum money on every single individual drug, do we? It'll be like my days back at Apple. When we came out with the Macintosh and sold 24,000 of them in the first year, I think that was the number, we didn't go, okay, cool, let's just ratchet up the price and then we'll make more money next year. No, we said, let's build more functionality into it. Let's get more software applications on it. Let's build on that platform to make it more useful and create value in more and more areas and sell more of them to more people, because this could be useful to every single person on Earth. This is the way gene and cell therapy is as well. It doesn't have to be the same game as the old pharmaceutical game where you happen to own a market because you have the only solution for this siloed, sort of disease area. And those patients are essentially hostages, right? And you decide how much they have to pay for it, and the price, the decision that the patient has is either buy your drug at the price that you put it out there at, or give up hope, die sometimes without that drug, right? That's the old pharmaceutical market, because it's non-competitive, because it costs $2.1 billion to make a new drug. Who's going to compete with you once you solve that problem in that siloed area? There's a natural barrier to entry. Gene and cell therapy isn't that way. Would it shock you, to your core, to hear that the amount of money that we spent on top of our platform development, so once we had the platform, once we had created the MS-DOS or the iPhone of diseases, to write the app on top of that to cure HIV cost us $15 million. That's enough money to get it through the first phase 1 trial that will get us safety and efficacy data. $15 million. For a traditional molecule, think $250 to $500 million for that. Remember, that's from discovery all the way out through the end of the phase 1. This is a gargantuan barrier to entry that is dropping because of the predictability, the reliability, the nature of developing drugs in this area that are about designing things instead of discovering things. It's not about generating 10,000 random molecules and figuring out which ones work and making a drug and commercializing it. No, you can sit down and you can assume that, hey, if Avexis can cure spinal muscular atrophy by replacing this gene in the CNS, if you have another gene and another disease in the CNS that you want to go after, hey, you can learn a lot from that project. It's not like a molecule, right? A molecule, what do you edit in a molecule to make it work differently? But a virus, right? It's got a vector inside, the software. Well, if that virus did a certain thing in the body and delivered genes to a certain place, okay, you can go in there and edit that little software. Instead of bringing in gene A, you can bring in gene B. And instead of firing it with specific promoter X, you can fire it with specific promoter Y. This is all very predictable. This is so much like software. Software is not a monopoly because it's so easy to develop. And you're going to see a lot of competition in pharmaceuticals that, just didn't exist in the past, to the benefit of patients, to the benefit of society, to the benefit of the world, and, by the way, to the benefit of pharma companies that can actually embrace this and do it. Why? Because pharmaceuticals are only $1 trillion a year right now. Even Goldman Sachs says it's going to be $5 trillion. Listen to me, it's $10 trillion. All right? They're making no money in this market. And why? Because they can't deliver enough value. Literally $1 trillion is all the world can afford to spend on the utility, the value that comes out of all of the drugs that are on Earth and available. But if these things start curing your cancers, if they start adding, years or even decades to your life, if they improve the quality of your life so that you get out of a wheelchair or you see again, think about the markets that are available, right? If I was a pharma company, all I'd be thinking about is gene and cell therapy. But I'm a gene and cell therapy company, all right? So I've got a platform that I think is going to enable this revolution. Today, what we're seeing is a drip, drip, drip of miracle cures coming over the top of the dam, all right? And then you're going to see a steady stream, and then you're going to see a deluge, and you can already see that it's accelerating, and then the dam's going to burst. So the pharma companies that live down below here had better be building their boats. And the excitement and the value that is about to be released is tremendous. It is an incredibly bright future that we all share. I hope that by listening to me, you recognize something that I recognize, that this is a revolution in pharmaceuticals and the future is bright for the world. And there's so much value that we can bring into this market that 1 trillion, 5 trillion, 10 trillion, we haven't even started yet. This is the software revolution for the next 100 years, creating value by writing important software for the human computer that improves health. What's the gist of this whole industry? We're converting deadly viruses. Does anybody doubt the power of a virus in the COVID era? We're converting deadly viruses. We crack them open, we scoop out their deadly software, and we throw it away. Now we can use this power to deliver things that make you better. We don't know everything that can be done yet, because to cure everything, we have to understand every mechanism of action in your body and every gene that relates to that. We're a long way from that, but there's a lot of low-hanging fruit already. There's a lot of monogenic diseases that can be cured. We're curing an infectious disease. We've got a great idea for cancer. And expecting an explosion, just a literal deluge of these things coming in the future. 7,000 monogenic diseases, thousands of infectious diseases, cancers. These things are all in our sights. All right. Well, thank you very much for listening. That will be the formal part of my presentation. I am happy to stick around as long as there's time allocated in this conference to take questions. Otherwise, we'll wrap it up. So thank you.
Yeah. So thank you so much, Jeff, for today's very extensive presentation. Everybody, I think we have received maximum reviews for your presentation today. Your presentation. A lot of people out there, and there are questions. There's so many questions. I'm just about to share it with you. Do you want me to share directly in the chat box, or do you want me to repeat it and then?
No. Just give me a question. Ask the ones that you think would be of the most interest to your audience, and I'll try to answer them if I can.
Okay. So one of the questions that I have received is, any plans to extend gene therapy of other human liver disease beyond PKU?
Absolutely. Okay. So the question is, are we thinking of other liver-centric diseases that we could treat from our platform? And yes, we absolutely are planning that. We have so much experience now putting lentiviral vectors into the liver, into liver cells, and we know exactly how it behaves, that we're not only going after liver cancer, but one of the reasons that we focused on liver cancer was because we had been working on PKU, and we had such a good understanding of what it takes to get into the liver and to reliably transduce the cells. So yes, liver is a focus of ours. It's not an exclusive area that we're going into, but it's a very convenient area. So the answer is absolutely yes. And as a matter of fact, because we're so good at the liver, if you could think of any monogenic diseases, metabolic disorders that are liver-centric, we're going to, in a few years, we'll be looking for collaborators. We can't solve every disease on earth ourselves. So what we're going to be looking for is experts, companies, doctors, researchers that are really passionate about a particular disease and really understand it well. And what else do you need to have? Models. If you have an animal model and a cell model, you've got something. And then if you've done enough experimentation in those models that you know exactly what the pathology of the disease is, and you can tell us what driver do we need to sever? What gene pathway do we need to sever? Or what do we need to replace or enhance? Okay, that's the birth of a collaboration. Let me tell you something else shocking in terms of pricing. If you came to us, you know how much it would cost us to give you experimental materials that reliably enhanced or severed a pathway in the liver that you could test in your models? Less than $100,000, and we could do it in less than 6 months. From start to finish. Could you imagine having a drug candidate that was worth testing in a mouse for a $100,000 investment? And at that point you could decide, or we could decide together, is it worth trying to progress this into the clinic based on the initial preclinical data that we get? And remember, the other part of the AGT platform is that we're very good at anticipating what are the problems you're going to see when you deliver this into human subjects. We've learned ways to test a lot of the potential problems at the bench where it's cheap to test, instead of testing it in the clinic where it's incredibly expensive to test. A failure in the clinic is really a disaster. It's really expensive. A failure at the bench, you wasted a few $1,000s. That's the idea. You would never write Excel, right? You wouldn't go write a competitive spreadsheet and without testing it, launch it, right? Of course, it'd be full of bugs, right? And by the way, if you found out it was full of bugs, would you throw it away and go, oh, okay, let's start from scratch and try that again? Let's write a whole new spreadsheet. We'll erase all that software and start from scratch and do it again? Of course not, right? That's not the nature of software. Software's debuggable. But that's the old pharma model. You have a molecule that doesn't work. What do you have? Practically nothing. Unless it just happens to have a side effect that's beneficial that you might want to leverage later, or you get a lucky situation where it turns out to be useful somewhere else. No, failure is like, that's the end of a molecule. A failure in gene and cell therapy is a matter of adjusting it, modifying it, debugging it. There's so many things in HIV that we can twist. If we don't get efficacy the first time, there's so many things that we can change, and we can bring that product back. Okay, very long answer to your question, but yes, liver-centric diseases are very convenient.
Okay, so I have one more question. One of our participants says that outstanding talk, and further, how about targeting the most prevalent human diseases on earth, beta thalassemia and sickle cell disease?
That's being done by great companies. Beta thalassemia is being researched by Bluebird. They actually use lentiviral vectors, and I believe that this is a high probability solution for beta thalassemia. Here's the problem that I think they might be experiencing at Bluebird, and I'm not qualified to comment on their clinical development, so Remember, this is just me talking right off the top of my head here. But I think one of the problems is that the old style of pharmaceuticals was if you found something that worked in the mouse, you rush into the clinic. If it works, you're rich. And if it doesn't work, you got nothing. All right, so they're caught in this weird purgatory right now, right? It worked in the mouse, so they went into the clinic, but it doesn't. Gene and cell therapies never really fail. They just have problems that can be fixed. And if you spend enough money on them in the clinic, you will eventually fix those problems. So look to my good friends and colleagues over at Bluebird to knock out beta thalassemia. They will do it. Hemophilia, there's a number of companies that are doing that. I don't think that, you asked sickle cell anemia. There's been a recent treatment of sickle cell anemia at the NIH with a drug that's developed, and I'm sorry, I can't remember the company that did it, but it worked. Okay, so, there's no question that these things are doable. If somebody else doesn't do them, I promise you AGT will. Why? Because we can catch up with a lot of these developments that are having problems in the clinics. We can catch up very, very quickly because we have this incredible tool set. Remember, they're trying to write software for the human computer without an operating system. Would you go out and buy one of these things with no operating system on it and try to develop an app? Of course not. It'd be super expensive. There'd be a ton of bugs, and it would be a lot of work to get it, out to market. All right, we're going to do that operating system. We'll use it internally if somebody else doesn't attract us to a project to collaborate. But that's the idea, is that we want to share that platform with the world, and we want to find a way to collaborate with anybody that has the energy, the focus, the passion, and the knowledge to cure a serious human disease. So if beta thalassemia isn't cured in 3 years, call me. All right? Because there's definitely a pathway there. And within 3 years, I think we'll be done with HIV. We'll be far enough into immuno-oncology. We'll have extra money in the bank. We'll have a lot of flexibility in terms of doing collaborations.
So, one of the question is actually related to the answer that you just shared. The participant says that yes, but stem cell transplant is not practical for most of the patients in the developing world. Still, they need SCT though.
Okay, so I'm really glad you asked that question, right? Because you're calling it a stem cell transplant, but it's in HIV, it's not a stem cell transplant. It doesn't have to be in any of these monogenic diseases of the bone marrow, right? Because you think about, where does hemophilia come from? It comes from a broken gene in your bone marrow that produces red blood cells that are missing factor VIII, right? And where does sickle cell anemia come from? It's basically the same idea. I'm not familiar with the root causes of all of these diseases, but they're fairly well understood. And the best technique that we have for fixing them right now is that autologous, cell therapy that might involve even stem cells that have to come out of the bone marrow and back into the bone marrow. And yes, I think if, your annual income in your city is, $10,000, you're not going to be able to afford the current cure for HIV or the current cure for beta thalassemia. But don't forget what I told you. Doubles in power every year, halves in cost. All right? Many people in those countries didn't have a telephone, and almost none of them had a computer, and now they all have one of these. Because that's the way technology works, right? We will find a way to cure you of HIV with a shot in the arm that has $1,000 worth of materials in it. Still expensive if your annual income is $10,000. But if the shot costs $3,000, you might still do it. You might be able to pull together that kind of money. And remember, a lot of the HIV that's being treated in Africa, for instance, it's not paid for by Africans, right? HIV is a global problem, and Africa is a place where there's a huge proliferation of this disease that's threatening to the entire world. So the Archdiocese, the World Health Organization, the US, the the Vatican, the European Union. They buy drugs from Indian companies that license technology from U.S. companies to cure HIV or to suppress HIV in sub-Saharan Africa. I think it's like $120 a year worth of pills, but it's some low number. Okay, so it's not out of reach, and I get it. Like the pharma model in the U.S. may not bring it out day one in sub-Saharan Africa or in third world markets. Capitalism provides an incredible incentive to do amazing things, but it can also be an impediment to the very important moral imperative of getting these cures out to people that really need them. So I can't promise you any miracles in terms of the time frame, but I guarantee you they're coming. I guarantee you that we can't help if we have to continue to be the number one provider of HIV cure. One part of our whole corporate culture is we don't rest on our laurels. All right, we cured HIV. We think we have a cure for HIV. We think we've got it already. Guess what our PhDs are doing right now? They're finding a way to cure it better, faster, cheaper. They already have something cooking in the lab that we think is 10 times better, that'll drop the cost by 50%. Right? How can we stay ahead of other companies if we don't innovate? Right? This is what I tell everybody in my company. If we don't continually work to obsolete ourselves, somebody else will gladly do it for us. That's the nature of competition. All right? So if somebody else doesn't come out with a better way than us and drive the cost down, we'll do it ourselves. All right? We don't have any choice. That's the beauty of this type of pharmaceutical. If somebody can develop a new drug for $15 million, like we just developed the last drug, who can enter this market? Anybody that's ambitious, right? Anybody that's committed, anybody that's passionate. All these other great attributes of human beings can now be applied to reducing suffering and reducing early death from serious human diseases. Our platform will enable that. We're happy to share it. Once it's established, once we get our company established, we're happy to collaborate with other people. So HIV, I predict, will be a shot in the arm like a vaccine, and one and done. You'll probably have to wait a few weeks for it to take full effect. Then you will throw away your meds, and you will be immune for life. All right? Now, I promise you that's coming. The only difficult part is saying exactly when, right? Because when I saw mini computers, I knew this was coming. I just couldn't tell you when, right? Because anybody that's a technologist sees the trends in technology. That's why science is so related to science fiction. What's a science fiction writer? Somebody who knows a science, who can just dream about what it could do in the future, right? And guess what they end up doing? They dream about things that come true, right? Star Trek, Gene Roddenberry, he wrote about you would have communicators in your pocket that you would flip open. Scotty? Yes, Captain. Right? What do we have today? What is this? Right? It is that dream that Gene Roddenberry had and put in that science fiction Star Trek. Right? All that stuff's coming. Came in computers, it's coming in medicine too. Dream with me. Believe me when I tell you all that stuff is coming. I can't tell you exactly when it'll arrive, but it will arrive.
Okay. So I have one more question from one of the participants. He says that how do you see the pharma versus biotech markets change with regards to patents? Now the definition of the drug is a gray area through decentralized versus centralized manufacturing, allowing localized hospitals with the right facility, allowing it to produce their own CGT products. What is now the defined intellectual property of CGT, the vector or the modified cell?
Well, it depends. So what will decide that? What's patentable, right? So we think the secret sauce is these reusable constructs that will become components in many different things. Think of us. Oh, we invented the transistor. And as a result, we're part of the transistor radio, right? Whoever invented the rheostat is part of that. Whoever invented the resistor, whoever invented the capacitor, the person who discovered radio waves, right? Patents will decide how the value of essentially, these products that are built from those components get divided up. But the value is associated with an invention, which is associated with a patent, which is associated with, having control of that over a certain period of time. All right? So decentralization, yeah. I mean, that's a very broad question. That's a great question, and I could spend 2 hours just on that question, and I have before. But yes, think about manufacturing for a moment. I think we're a cutting-edge gene and cell therapy drug development company. Guess how much manufacturing we have? 0. Why? Because manufacturing is not high-tech, in my mind, right? Anybody can make a lentiviral vector that has money to put together bioreactors and, and purifiers and filtering systems and all that stuff, right? That is not rocket science. Rocket science is, what do you tweak inside your body that cures a disease? That is complicated. But once you've figured that part out and you've made the plasmids to make the viral particle, there's about 40 companies that we can go to that will make the viral particle for us. It's not as cheap as us having our own manufacturing, but it's like in our current stage, we couldn't keep our own manufacturing busy. So why would we build a $10 million facility that we can only use 1 week a year, when we can rent 1 week in somebody else's $10 million facility, and all we have to pay them is the equivalent to 3 weeks, right? So it costs us 3 times as much as if we made it ourselves. But look at that. It's like 1/15 the cost of, the annual cost of making these things. So yeah, manufacturing, this is now a layered value proposition all the way from the actual drug down into manufacturing, up into developing actual applications that you use these drugs for, to the delivery systems, to everything. Look, I think ultimately, this is going to sound crazy. Do I have enough time to go on a little bit longer? Because I see you tipping your head. But here's here's how I think it'll ultimately happen.
All the queries, actually. I'm just checking all the queries that we've received.
Oh, okay. So let me show you this. Can you imagine a day when this cell phone, you just do this, and it reads your DNA, all right? And it goes into a central facility that goes ahead and compares your DNA to, let's say, 1 billion other genomes that it has, and it figures out why you're getting the symptoms that you're getting. And then it sends back by software the instructions for a replicating virus to your cell phone. Your cell phone synthesizes the virus, and you do this. And a week later, it's spread around your body and then expired. But in the meantime, it's changed the defect that was driving your pain, driving your disease. It's not impossible. It's science fiction today. It may be science fact in 150 years, or in 100 years, or in 50 years. I don't know. A very interesting movie. I'm always bringing this movie up. Elysium. It's a Matt Damon movie where all the rich people live in a satellite circling the planet. And they have a machine which, for some reason, they don't share with people that are down on Earth who have to repair robots for a living. But what the machine does is you lie down in it, and you see this thing scan over your body, and then it goes over your body one more time, and it cures whatever you have. Oh, you have leukemia. You're cured. Is that really impossible? You tell me, is it going to be impossible to rewrite DNA with electromagnetic waves? I don't know. We may be able to find ways to scan your DNA without touching you, and to rewrite your DNA reliably in your entire body without touching you electromagnetically. If that's the case, Elysium is not science fiction. It's a prediction of the future, and that machine may one day exist. So anyway, that's that's what I think about. I won't be alive to see those things, right? I may, through gene and cell therapy, I might live to be 160. If I am the CEO of a really successful gene and cell therapy company, I may be able to afford all the things that, allow me to live a very, very long life. That will be another sad factor, by the way, in gene and cell therapy, is it'll open up a new gap between the haves and the have-nots, and I'm not excited about that. But that's the reality, is that, some of these things are going to extend your life by virtue of getting rid of all the things that tend to kill you early. But we already know that if you have heart disease and your heart is giving out, that we are very, very close to being able to 3D print a structure for a new heart and populate it with your heart cells, regrow your heart, replace your heart like it was a water pump on your car, right? And that's the point, is that, regenerative medicine is just the extension of gene and cell therapy, and regenerative medicine will lead to replacing, it seems possible to do this in the relative near term, in the next 30 to 50 or 60 years, on all your organs except for your brain. And the only reason is we don't understand how your brain works in terms of your memory and your personality, right? But once we understood that and could record it and put it into a new organ, which could happen too, we'll be able to regrow your brain also. Then you're just going to be like an old car, and we can keep you going as long as you can afford new parts. Amazing. Scary and amazing.
Okay, so I have one more question from one of the participants again. Do gamma delta cells overcome immune privilege as seen in many organs? By the way, it's a fantastic talk. That's what he mentions.
Thank you. Thank you for the compliment. So gamma delta T cells tend to ignore checkpoint molecules. If that's the question, they do not get turned away by checkpoint molecules. Yes, that's our experience. So they will run right past it. You won't need to take Keytruda first so that the gamma delta T cells can see the tumor. No, all you need to do is light it up with our viral vector, and that will be enough to cause this catalytic reaction in your body that clears the epithelial malignancy.
Okay, so my next question is, how do you see, how do you foresee patient monitoring in these personalized medicine?
I see the patient monitoring being very, very long in the first experiments, right? Because what are we doing to you? We're permanently rewriting the software of your soul, right? A tiny little mistake might not come out for 20 years, right? So we are required to monitor our patients for 15 years. And it makes total sense, right? Now, the other thing that the FDA is doing, and I don't know, I can't speak for all the regulatory bodies worldwide, but the FDA is doing a very clever calculation where they're saying, okay, given that it might have this long-term danger, let's restrict the use of these things to really acute needs, right? If somebody's going to die, it's worth the risk. How would you feel if you had a deadly cancer where the doctor said, it is almost a sure bet that you will be dead within a year? And then I brought you a treatment that gave you 10 years of life, and at the end of 10 years, you exploded with a new type of cancer never seen on Earth before, where you had literally, a week to say goodbye. I bet you in that week, you'd write me a letter and thank me for the extra 10 years, right? Because that's the point. If it's really, really serious, it's worth the risk. Now, remember, everything we learn from monitoring these patients for 5, 10, 15, 20 years, we can put back into the debugging process, and pretty soon the FDA and other regulatory agencies will understand so well what the implications are of each change that we make that safety testing will get smaller and smaller and smaller because everything will become more and more predictable, right? And so, and also 15 years, they'll be like, you don't need to monitor them for 15 years. We already learned from all these other drugs that that drug is totally gone within 5 years. So monitor them for 5 years, right? So it's all going to be shifting, but it's all going to be shifting in a positive direction that lowers the cost of all these things. And by the way, if you're a doctor asking that question, you will not go out of business. There will be more and more things to cure. There'll be lots of patients to see. Don't worry about the follow-ups on clinical trials. There's going to be so many clinical trials. You'll be glad to get rid of the patient faster so you can bring in a bunch of new patients and share in the excitement of really making a difference in people's lives. That's what motivates us at AGT, is that excitement. And I know it motivates, physicians globally.
Thank you. And so one of the next question is also one of these issues with autologous CGD is the huge upfront cost due to high production cost, but low monitoring cost down the road. How do we overcome the politics of these costs? And for instance, with HIV, antiretrovirals have a cost of 50,000 USD a year on NHS in UK. However, CGT would cost close to $300,000. How do we fund this with such a high HIV plus positive population? Would you foresee an off-the-shelf allogenic move in CGT? It's quite a big question.
It's okay, so it's a good question. I understand what he's asking or she's asking. So let me unpack it a little bit. First of all, I get it. Antiretroviral therapy is only cost $50,000 a year in the UK. But what you're forgetting is these people that are taking antiretroviral therapy, it's a toxic chemotherapeutic. Forget about the fact that it makes their life crappy every day, all right? Nausea, diarrhea, headaches, fatigue, loginess, dizziness. Long-term exposure to antiretroviral therapy leads to early aging, bone density issues, osteoporosis, liver, kidney, heart disease, extra cancers. Guess how much it costs to maintain an HIV patient per year in the United States, with the drugs costing, the drugs are $20,000 to $30,000 a month in the United States. So let's call it $300,000 a year in drugs, all right? I'm sorry, that was a year, sorry. Let's call it $30,000 a year in drugs to treat HIV. We're spending another $50,000 to $100,000 a year on every single patient treating the comorbidities of the toxic antiretroviral therapy that they're taking on a daily basis. These patients don't cost $50,000 a year or $30,000 a year or whatever. No, these patients cost $100,000 a year on average, and they're infecting other people if they don't religiously take their antiretroviral therapy. And some people won't even take it because it's so painful, and so they're out infecting other people. So when you start adding up the societal cost of this, $300,000 to end that person's suffering, to end that person's endless doctor's visits for early aging, bone density issues, osteoporosis, liver, kidney, heart disease, cancer, and just feeling like crap, to restore their productivity, holy cow. Gene and cell therapy is a bargain already. And don't forget, the head of NICE said that CAR-T, remember CAR-Ts, $400,000 to $500,000 to cure a formerly incurable leukemia. You know what he said about him? It'd be worth it at twice the price. Is he crazy? No, because dying isn't free, right? When somebody has a terminal leukemia, they don't go, oh, thanks for the news, doctor, and die. No. We throw everything we can at them, and we extend their life out to a year, and it costs us $1 million, and then they still need to buy a funeral. Right? I mean, it's ridiculous. And that you can cure 60% of them for half of that price? It's a bargain. That guy's right. He's not crazy. He's absolutely right. The question comes from a place of just looking at the drug cost, not the cost to society, not the cost to the patient, not the alternative cost, right, of everything that goes into that patient who's got that disease. And sometimes just being a little bit too caught up in the pharmaceutical model, right? Pharmaceuticals are a very isolated thing where they justify their pricing in sort of weird economic ways that tend to tilt the conversation, about pharmaceuticals, maybe in a way that ignores the elephant in the room. The elephant in the room is the patient. How does the patient feel, right? What's the value we're giving to the patient? Trust me, the future of pharmaceuticals you'll be glad to pay for them because you literally will pay less money for the drug than for what you get in return. Why, by the way, don't we write letters to our congressmen and our politicians about, hey, cell phones are expensive, regulate them, make them charge less money for these cell phones, right? We don't do that because cell phones, relative to the value that they give, are okay. Yeah, they're expensive. Believe me, Apple can sell you that new iPhone for half the price and still make plenty of money. They can sell it to you for a quarter of the price and still make plenty of money. So can Samsung. All right? But we don't deny them some profit for the innovation and for the fact that they keep giving us new cell phones every year that are even better because the value is there. But if you take Avastin, I'm going to pick on Roche here for a second. Forgive me if there's any Roche people in the audience, but Avastin, it is prescribed for terminal cancer. It costs about $125,000. You'll spend about 12 days in an infusion chair getting it, all right? And how much longer will you live on average? Unfortunately, I can't see you guys in the audience, but when I ask this question, people are like, 6 months, right? They think they're being given a short one. People say 3 years, 2 years. The answer is 6 weeks extra overall survival for $125,000. If they could guarantee you that was the outcome, what would you do? You'd call up your insurance company and go, look, I got a bargain for you. Give me $30,000 and I won't take it. I'll just go on vacation for the time I have left with my family and enjoy my last days, right? Because it makes no sense to take Avastin. But why do you take it anyway? Because you turn around, first of all, it is approved. It was approved in the old days where if it showed significance, that was good enough, right? So now, the doctor knows if he doesn't mention it to you, what will happen? In the United States, I don't know what will happen in Europe or in India, but in the United States, the doctor doesn't mention Avastin because he knows it's not really that great an idea. You could even get side effects. You're going to spend 2 of your 6 weeks in an infusion chair, right? He's going to say, look, it's really not worth the trouble, because there's not that much variation. But you're going to say, well, I know 6 weeks is average, but, is it possible I'll get longer? Because we're hopeful, faithful creatures. We would not have lived long enough as a species and been this successful that we dominate the planet if we didn't cling to life, if we didn't claw our way back, if we didn't fight tigers when we got cornered, right? We evolved to cling to life. Most animals are that way, but we are really good at it, including rationalizing when the doctor says, well, could be longer. What do you hear? All right, I could go longer. Well, what's the longest you've ever seen? Well, I had a patient that lasted 2 years after the treatment. All right, now you're thinking, if I get 2 years, something else might come along, right? We're hopeful, faithful creatures. Avastin is $7 billion a year of hope. Hope is something that's worth money to us, all right? And look, I don't mean to denigrate Avastin either, but what I will say, and I'll say this straight to Roche, Avastin's going to be replaced by something that has an extra overall survival of 5 years on average. Nobody's going to buy Avastin for $125,000 when you can get that drug for $300,000. They will sell their house if they have to. Because would you rather drive a fancy car, or would you rather be alive to drive a crappy car? All right? It changes the whole equation when you finally give that kind of value. And that's the way the drug industry will be. People will choose their drugs based on who's giving them the best value in return. And the cost of the drugs will be less than the value they bring you. They will be like cell phones, where you're like, it's expensive and painful, but I'm going to buy one anyway because I love what it gives me. Any other questions?
Yes, I have one more question. I think, you have done all, you've given it all. You've been very energetic throughout the session and very enthusiastic as well. I don't want to, share much more queries and want to burden you anymore. I think, just one more question for you, and then we can done. We can be.
All right, we'll make this the last one. Sounds great.
Yes.
Thank you.
Yes.
It was a pleasure, by the way. So I enjoy this. I think this. Look. The reason that I'm in this market, I was retired before I decided to start this drug company, and I risked my whole retirement on this. Now it's going well now, and I'll probably retire much better than I was back in my 40s. But I did it because I'm passionate about what this can bring to people. I mean, think about the joy that everybody at AGT would get by curing HIV, by making that kind of difference in other people's lives. That is something that humans can derive incredible pleasure from. This is more than a paycheck to work at AGT. It is an opportunity to make a difference for your fellow human beings, and we cherish that. So that's where my passion comes from. That's where my energy comes from. That's where my enjoyment to get out there and talk to people comes from. Because, this is a revolution, okay? There it is, revolution. I even said it. You're all part of it now. You are starting to get a sense for what the future might be. You are creating demand for that future. You are creating a sort of expectation of that future. That brings that future faster. All right? So, yeah. So anyway, don't worry about, it was 2 hours. It was 2 hours of fun for me. I appreciate the opportunity.
For us as well. Absolutely. It was pleasure for us to, even to have you on the screen and to hear so many things. I think, you gave so many difficult, theories in quite simple way possible so that, a layman can understand, I would say. So it was easy for us to understand. Though we are not from, your background, we don't understand much. But yes, we could understand that. So thank you. So good job, great job, I would say. And, coming back to the last question, I would say that how do you raise funding to do all this amazing work? Venture capital or big pharmas?
Okay, I was afraid it was going to be a question that I couldn't answer. That would be hilarious. The last question, I'd be like, I don't know, right? But I do know how I raise the money. Okay, so AGT bucks tradition in every single way, all right? It was only the fact that I came from a totally different industry, that I had a vision for this industry that other people within this industry didn't have. There were lots of biotech people. There were lots of scientists that understood viral vectors. There was companies that were doing gene and cell therapy before I came on the scene. But what I saw was sort of a unique perspective on it, because I came from something that wasn't all just hardened or, sort of forged in the traditional biotech market, right? I could think outside of the box. As a matter of fact, I couldn't do anything but think outside of the box. Okay, so I established this model, and it was a vision that just welled up inside of me about the future of pharmaceuticals. And, it just made sense to me so much that I poured my own money into this. The first 8 years were me and my family pouring out our hard-earned, money. Me and my wife literally risking our entire retirement. That took about $2.5 million. Plus, guess who had a big belief in gene and cell therapy? NIH. So they kept giving us grants to try it on this and try it on that and try it on that. And we were doing all these studies, and that added together, we spent about $4 million of our money and the government's money, and we had developed a whole bunch of components that were likely to be of value in future drugs, that operating system, right? And the way we develop the operating system is just keep trying diseases and figuring out what's in common between all of them, and that's our intellectual property that's come to be valued in a formal valuation at $2.4 billion. All right. So luckily, and sometimes it's not just skill or vision or whatever, but around that time, around 2012, 2013, there was a rebirth in gene and cell therapy. People were starting to think, maybe this thing isn't such a dumb idea after all. Because you may remember that there were some accidents in the late 90s and early 2000s that sort of threw a shade, threw a pall over the entire industry. And so, venture capitalists, no interest. Pharmaceutical companies dumped their projects, stopped investing in it. No strategics, the whole traditional industry turned away from it. That's right when I came into it, 2006, right? But I was looking at the fundamental elements and saying, yeah, I see from here to there, right? I see a day where gene therapy is so big on Wall Street, it's like the .com boom. Like literally, if you have gene in your name, you're getting phone calls from investment bankers. I want to take you public, and it'll even work, right? Because that's what the .com boom was. Finally, people realized the internet was big, and everybody's making money on the internet, and then Wall Street was like, hey, IPOs are easy. All right, let's call up everybody that's got internet in their name or .com in their name, and let's take them public. We don't care what they do. We only care about whether we can issue an IPO, collect a commission, and that it's a good transaction. They get their money, we get our commission, and then, okay, hopefully it'll work out for everybody, right? Because that's what Wall Street does. It can be very, very powerful. It raises lots of money when there's interest. But the point is they weren't back, but at least there was some level of interest in the investor community. So I looked at myself and I said, there's got to be other people like me that understand, can embrace this vision, and would be interested in risking some of their money on this. I don't want them to risk their whole retirement, but would they give me, say, 1% of their equities portfolio? If they had $10 million in the stock market, could I convince them to give me 1% of that? $100,000 to swing for the fences in something that might turn $10 into $10,000, right? Because I could see that future. And so what I did is I went out and I did something that I learned from when I was at Apple. It's called evangelism. I went out there and I said, I'll talk to anybody that will listen to my great idea. I believe this idea to the core. Evangelism is associated with religion, right? But the point is it's how do you make an emotional connection with somebody so that they see your truth? It's like an art form. And they can connect emotionally to your journey, to your vision, in such a way that they might join you in that journey, in that mission. So I went all over the country. Anybody that would listen to me, I'd talk to them. And high net worth individuals like myself started putting in money. And in 2013, we raised $1 million. That was the first outside money. It was the first time that I could look my wife in the face and tell her, I think we may not lose our retirement over this. I think we may have turned a corner here. Other people are interested in what we've created. And then, a year after that, we raised $3 million. A year after that, $4 million. A year after that, 15, 16, or 17 million. A year after that, we were raising $25 million. The valuation went from 15 cents a share, which would be what you would call a company valuation of about $4 million pre-money. The next year, it was $15 million pre-money. The year after that, $48 million pre-money. The year after that, $120 million pre-money. And our current round at $25 million raise, and we're starting to have, sort of angel funds come in and family funds and whatever. the valuation on today's round, 240 million pre. But if I show first human efficacy in just HIV, I can sell that project alone to a pharma company for a $15 billion check, $300 a share. I don't even have to sell the whole company. I just sell them that project, and I will get a check that I can divide between those investors that are paying $5 a share in this round, and I could return $300 a share. Now, keep in mind, I've gone off into investment stuff, and the law says I must disclaim everything that I'm saying. I'm talking about future performance that cannot be guaranteed. This is not a solicitation, all those things. So let me disclaim everything officially. But let me say, this is what I believe is going to happen to me. I'm a big shareholder in this company. This is what I go out and I tell what are called accredited investors. Accredited investors have the net worth or experience in investing that they can evaluate these things realistically so that I'm not cheating them out of their money. They can look at, they can do the due diligence. And we've got plenty of data. They bring scientific experts sometimes. You never know what they're going to do. Sometimes they're risking so little money, and as far as their portfolio goes, they're just like, gee, Jeff, you seem to believe it. And I'm all excited. Here's a check. That happens sometimes, right? But it added up to what? I think we're at like $44 million so far. Now, it's miraculous what we've done on $44 million. And we have a valuation report on our patents. So if we go out of business, we can still sell off our patents, right? We may not get the amount that they're valued at, $2.4 billion. But the guy who did the valuation said we can get half of that, $1.2 billion. Guess how much that is per share? People are buying shares at $5 right now. $1.2 billion is roughly $24 a share for going out of business. All right, but I took half of that because if I'm going to sleep at night, if I'm going to go out there and I'm going to tell people what I believe and feel like I don't want to accidentally even lie to people. I don't want to be so caught up in my own thing that I might be saying something to somebody that I would feel shame about later, right? And so I look at it and I go, all right, let's take half of what the lawyer said, $600 million. That's $12 a share. They're paying $5 a share. I'm feeling fairly comfortable that we have insurance that is unlikely that they're going to lose their money. And in fact, probably the worst they can do is double their money, right? Okay. And for that, they're getting a free lottery ticket to the big game. Because if I cure HIV, I won't give them $10 a share. I might ask them to buy back their stock at $300 a share. And I might recommend, hey, only sell back half your stock. Play the rest of the way through on house money. So what happened to VCs along the way? You think, oh, VCs like to make money. No, they don't care about making money. They care about managing money. They make 2% from managing a fund. So they have this fund here, and if they can show success in that fund, they can raise a bigger fund 2 years later. That's how they give themselves a raise. 2% of the money under management is their salary. And the only thing they don't want to do is have a failure in a fund. So what they do is they learn a very narrow technology. They found a place where they know how to make money, that they understand, that they can pick enough winners to make how much? What does the average VC fund return? It's like 12% internal rate of return, IRR. 12% per year. If you're a billionaire, 12% is pretty good on your money. That will keep you in, tea times and jet fuel and whatever. So VCs are, out pursuing this money. And what they're saying is like, look at our track record. Fund 1, 15%. Fund 2, 13%. Fund 3, 30% IRR. Here's our goal, right? So they're not attracted by 60x. What they're attracted by is that it fits their model. Now, what's another part of their model? If they have to show a profit in this fund, they have to show it by the time they raise this fund. And so what they do is they need to control you so they liquidate you by the time this fund rolls around. Because they only have a limited handful of companies that have to refill this bank account plus 12% per year, compounded, right? And so they need to make sure that as many of them are under their control so that if some ambitious CEO wants to save the world, they can stop him. because he may not be willing to sell out his company on the schedule that makes sense for them. So they always buy a third of the company. Well, I can't say always, but frequently they go, here's a company that fits our model. They buy a third of it in the first investment. It's them and a couple of other VCs. Why a couple other VCs? A lot of VCs are scared to make decisions on their own. They want to spread the risk so at least they can go, well, those guys were idiots too. It wasn't just us that got fooled by that guy. We got fooled together, right? It's risk management for their reputation. So they always come in groups, all right? Then they try to invest enough money that they own 35% of your company day one, right? And you're all happy because you get a giant check. It's usually more money than you need. But what comes with is they get a board position. And they're on the board going, spend, 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 faster, 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 time is money. And they make sure that you run out about a year later. Now you need more money. If you haven't hit some milestone where you can just drop those guys and go to new guys, guess what they own in the second round? 51% or more, right? Now they have what they need, right? If you're a decent company and they have absolute control, then this is the best thing for the model where you only care about getting 12% and you don't care about changing the world, saving the world, or doing anything that is just revolutionary, right? So the days of the old VCs where it was just like you meet somebody like Mike Markkula in your garage and he's like, I like your idea. Here's $250,000. Let's do it, right? And he adopts you. Steve Wozniak and Steve Jobs, they were like adopted children to this guy, right? And he takes you to the trade shows, and when you need money, he gives it to you. And when you can't get along with the CEO, he fires the CEO and he steps in temporarily himself. And he reminds you to wear shoes at Comdex, right? Those days are gone. Nobody takes risks anymore in the VC world, in the crossover investment world, in the investment banking world. No, you don't need to play the game if you control the game, all right? So we are outsiders. If you're a high net worth individual, you might go, yeah, okay, I'll put most of my money with those guys because Goldman Sachs always managed to get me 10% per year. All right. But you might say, I could spare $100,000. The average investment in our company, I think, is $170,000. We have some people that write $1 million checks, $2 million checks, whatever. We have some people that are in for $25,000 that in the earlier rounds, we used to take small investments like that. So, and we're able to live within reasonable money. Look, we can't do everything that we want, but we can contain everything. We can stay within the amount of money that we can raise this way and do amazing stuff. We are on the path to creating a revolution, supporting that revolution, being a leader in that revolution. I'm highly confident that we'll do that. And I don't know, in the future, if we cure HIV, raising money won't be a problem, right? If we want to commercialize that, 40 million people on Earth have that disease. Tons of really high net worth individuals in the United States have that disease. Literally, there will be people lining up to be in our clinical trials who are super rich people, right? So, money won't be a problem when we cross that threshold, and I'm hoping we'll cross that threshold in the next 12 months, but we'll see. But I think it's, I think it is this passion that you're seeing. It's this true belief in what I am doing. It is 12 years of watching this trend and knowing that the science fiction is turning into science that gives me the level of confidence that allows me to go out there and story tell, right? I am evangelizing my vision, my mission for the world. And some people find that incredibly attractive, incredibly engaging. And what do? This is what evangelists do. When they find believers, those believers join and they power that mission. They power that vision. And that is what is going on. And that's how I raise the money. So no, nobody came over and gave me a big check. Pharma companies, I talk to pharma companies all the time. I've got things that would make them so much money. They have opportunities to grab them now while they could get them at a reasonable price. And they're like, well, that doesn't fit our model. They just go, it's too risky. Right? They don't understand how fast gene and cell therapy is coming on. And they could be surprised. When I was at Apple, IBM just turned up their nose at us. Right? They just thought we were nothing. Apple was, I think, at one point, they offered to sell Apple to IBM for $100 million. Where's IBM now? They install personal computers for a living. They thought personal computers were toys when they met Apple. But Apple had a different vision, which said, these toys are going to become more and more powerful, and they're going to be in every house in the world. They can bring value to anyone. And where's IBM? That's one good question. But I'll give you an even better question. Where's Burroughs? And you're going, who's Burroughs? Well, that's my point. They were the second biggest mainframe manufacturer in the world. Gone. Sperry Univac, gone. RCA made mainframes, gone. GE had a mainframe division, gone. And I'm not even mentioning Digital Equipment Corporation, mini computers, gone, 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 gone. Right? Why? Because some people can see a future and can see a path to that future, and they can get just enough support to make that journey. And nature will determine what is real. If you are watching COVID and you don't believe that, you're missing the point of COVID. Doesn't matter how much politicians say, don't worry about it, it'll never go anywhere, I'm doing everything. Doesn't matter. The reality will be determined by nature. Gene and cell therapy is a force of nature. I didn't invent anything here. I just saw the power. I don't even do the work in the lab. I just told my vision to a bunch of scientists and they went, that makes sense, and they joined me. I told my vision to a bunch of investors. They said, that makes sense, and they joined me. They weren't traditional investors. The bigger the organization, the more successful the organization, the less likely they are and the less able they are to embrace a disruptive technology. That's why Burroughs ignored the microcomputer. They were victims of their own success. I can't argue with Roche or Pfizer or ViiV or Gilead about the nature of success in the drug market. They have it all, and I have none of it. I don't have a single drug. But if I cure HIV, it might be worth a second look at that point, wouldn't it? That'd be a big thing. And if they then see, oh, and they got a cure for cancer right behind that, that might raise a few eyebrows around those same companies. But then the problem is this: how fast can they turn that giant battleship? Because every organization is also an organism. It is made up of cells called human beings, sitting in cubes and doing their job. And what happens is that the organization gets a DNA. A DNA that's hard to fight. They have a standard way of processing things. I've had biz dev people from major pharma companies tell me curing HIV makes no sense from a financial standpoint. That's ridiculous. It's ridiculous. If Gilead buys our HIV cure, their antiretroviral therapy revenue will go up by double immediately. Now, why would that be? Are you going to take J&J's antiretroviral therapy that you have to take for life? Or are you going to buy Gilead's that you only have to take for 3 years and then you can be cured? Duh. Of course. It's an easy decision. But here's the kicker. Only 1 in 4 people that knows they have HIV in the United States, Europe, and other insured markets is taking antiretroviral therapy. 1 in 4. That means there's three times the market that is not taking your drug. Why? Because it's painful, and some people don't even expect to live 10 years. If you're an intravenous drug abuser, you might be thinking, I'm not looking for something that's going to make me feel sick every day, this toxic chemotherapy. I'm looking for my next high. I'm not going to spend money on that or time on that. I'm going to die within 10 years anyway. So you remain contagious. And, you know, as a result, the United States gets 50,000 new cases per year. And, you know, that's a lot of it is from, you know, people that think that they don't have much of a life, and so they don't treat their disease. But there's also a bunch of people that are just putting it off as long as possible, because the reality of it is this. If you find out you have HIV, and I'm not recommending this, by the way, so very careful. You find out you have HIV because you have a blood test, not because you go into the hospital with pneumonia, right? But you have a blood test for insurance purposes or something like that. You find out you have HIV. Then they put you on antiretrovirals, and you go from feeling normal and good to feeling like crap every day. And then you ask your doctor, how long is this going to be? And he says, forever. Then your next question is logically, do I need to take these drugs right away? I mean, I only found this out randomly. Could I put this off for a few years? Because it would make sense. If you could have 3 normal years and then suffer for the rest of your life, is that greater than, you know, suffering for the rest of your life? Yes, it is. So it's almost logical. It's irresponsible, right? You're contagious, right? But what are you going to do? You're human. You'll rationalize. You'll go, look, I'll be careful. I won't give it to anybody I love, or anybody I don't love, for that matter. I'll be careful. You know, if you really can be that careful, it's not that ridiculous a decision. But what if you find out instead, oh, you've got HIV. We need you to go on antiretrovirals for 1 to 3 years, and then we're going to give you a drug, and HIV will be out of your life forever. You don't even have to worry about HIV anymore. Like, literally, you can't get it back. You'd be like, sign me up, doc, right? Then you feel like crap. But you hear, oh, 1 to 3 years. I can do it. I'm going to have to do it someday. I can put it off for 3 years and then 1 to 3 years. But guess what? Guess what the downside is at that point of putting it off? You might damage your immune system so badly from untreated HIV that you may no longer be curable. Because remember that cell therapy? We have to find HIV T cells, and you have to be capable of supporting them in your body when we put them back in. Your lymph nodes need to be healthy. At some point, HIV will ravage your whole body. It won't just kill your immune system, it'll kill your lymph nodes. It'll kill critical parts of the immune system that may be difficult or even impossible to recover. So it turns the logic on its head. So Gilead should be partnering up with us and trying to get this out as fast as possible. And so should ViiV, and so should, who else is in this market? Sanofi and Johnson & Johnson. But what they believe is that, no, a cure will be like hepatitis C. You cure all the patients and the market goes away. HIV is way better than that. Why would we give it up? Right? Gilead's making $16 billion a year in ART. They announced the cure. All right? If they helped us rush this to a result where we could hold it up and they could say, yeah, we partnered with AGT, they made the cure, and we own it now, their antiretroviral therapy would double immediately. Why? Because remember those 3 people that aren't being treated? One of them would come in. That's a doubling. And where are they going to go? Are they going to go to J&J where they take ART for life? Or are they going to go to Gilead where they take it for 1 to 3 years? It's simple. They should be rushing this to the finish line. But they're not, because they're worried about that the current pharma model says that lifetime treatments are more profitable than cures. And let's just compete for the lifetime treatment market, because that'll be here forever. But it really won't. You know, there will eventually be a cure for HIV. They won't even look at our data rooms. It's too risky, right? We go, hey, we have proof in our data room. If you don't look at our website and believe that those people would have never signed up to be on our scientific advisory board, clinicians, all that stuff, if this wasn't for real, if you don't believe the article in Molecular Therapy from National Institute of Allergies and Infectious Disease that this stuff is that potent, right? Go look at the raw data. You'll be convinced. They're like, that's too risky. if we read your data room, what if there's something in there that you sue us for later because somehow, we were working on something powerful? We need deniability, right? I mean, it's like, it's too risky for them to even look at this. That is the DNA of pharma companies. They are incredibly successful. They are absolutely right about their models within the traditional drug development industry. And the only thing that I have to say to them is gene and cell therapy is a disruptive technology that changes all the assumptions that you've based all your models on. And the earlier you think about this, the better. You may be the IBM that survives the onslaught of microcomputers instead of the Burroughs, Sperry Univac, Control Data, RCA, and GE, right? Some pharma company out there is going to figure out how to embrace this. It's going to be a wrenching experience. You better believe that IBM, Lou Gershon, look up the case. It's a Harvard business case. I mean, Lou Gershon had to go to his executive staff and his board members and just say, hey, remember what we used to make all this money on and dominate? It's gone. It's a goner. It ain't going to be here. What can IBM be after the disruption? And he said, here's what we can be. The most trusted, biggest consulting company in the world that delivers great solutions of PCs for business. And they are still here. How many mainframes do they sell every year? I think it's 0. Watson is just a PR thing. It may do something great. Supercomputers may have some value. I think they will. But think about even a supercomputer. Can it really compete with all of the computing power attached to the internet? If you could borrow all of those PCs at night when they're not being used, couldn't they do as much or more than Watson because they have more computing power? Look, gene and cell therapy is the future. I'm certain of that. And I hope I've convinced some of you of that. I've given you some perspective on that you could, if you believed it already, maybe you have more reason to believe it, or you have some solid information that would sort of give you additional perspective on this. And I hope that that's what I've been able to bring here today. And, but the traditionalists aren't going to help us. They're stuck. It's not their fault, right? They've been successful for 30 years or 50 years. They're not going to risk everything if they don't have to. One day they'll realize that they may have to make a move if what I'm saying is true. I believe what I'm saying is true. They would argue with me. You decide for yourself. And then we will see, because time will tell. Nature will decide what works and what doesn't work, and whether gene and cell therapy brings all the miracle cures that I see in our future, or it doesn't, and whether it disrupts everything you understand about pharmaceuticals, or it doesn't. All right. Thank you.
All right. Thank you. Thank you so much. And thank you for being the star speaker of the day. It's truly amazing to have you on board. And, I think everyone has something or the other to take away something from you. It was undoubtedly the last session for 2 days even, but the best one, I would say. So it's a compliment from not me, but also the other viewers as well. And
thank you.
Yeah, thank you.
That's it's a huge compliment, and I really appreciate it. Thank you so much for the encouragement. And I'm glad it worked out well for you guys, and it was a pleasure for me. Thank you.
Absolutely, absolutely. And with this session, I would like to thank all our speakers and the attendees as well. So when I talk about speakers, I would like to mention special thanks to Dr. Mohammed Tutunji, Dr. Guillem Modol Caballero, Ms. Kirit Kaur, Dr. Maisha Rashid, Dr. Arun Srivastav, Dr. Sridhar Selvaraj, Dr. Michael Schenk, and, Dr. Govind Kokaya and Miss Helen Gu, and also, our last speaker of the day, Mr. Jeff Galvin. Thank you very much. It was amazing. I would say I was starstruck throughout the session. I just kept looking at you and the way you explained things. It was amazing. We enjoyed, and I think my words wouldn't do much justice today if I think I might go on and on. That's what me and I think my colleagues also felt the same thing who are watching you right now from Databridge. We are completely honored to have you on board, and we would like to have you next time as well.
Thanks. I'm happy to come back again. And please get into our website, americangene.com. Sign up for our newsletters if you want to track this. There will also be invitations that come out every once in a while, because sometimes we'll just do, sort of lunch and learns and whatever. So if you're interested in the sort of things that we talk about here, stay in touch. All right. Follow us on social media. And, I hope I'll see all of you again.
Yes, stay in touch and, get connected to, American Gene Technologies. They have a long way to go, and even you have something to gain for. So why not sign up their newsletters and go for it, I would say.
Fantastic.
Thank you. So with this, we are ending the live session. Stay connected. Just hold on. I think, so we are ending the live session. I've informed. Yeah. So yes, off the record now.
All right.
Finally, it's done. Okay. So, I just wanted to understand any feedback for us that could be more helpful and get your onboarding process more easier next time. Anything for us? So, what we can do for you? Any feedback?
This was very smooth. It was very easy. We happen to have a very sophisticated setup here. I don't know if you saw Norman at all. I guess you probably talked to him earlier. But he and our IT guy, Hamani, designed a system that allows me to see what I'm saying almost real time, even though there was a big delay on the live stream, and also see you real time, which was really helpful to see your smiling face. And, it's much harder to talk to just a camera than even just one face makes a big difference. So it's very easy, and apparently whatever system you used interfaced beautifully with what they designed. And so we were able to mix the slides in the background and, and do our standard type of presentation that we always do online. And we were able to accommodate your system. So no suggestions whatsoever. Just keep doing what you're doing and invite me back, please.
We would love to, absolutely. And, thank you for staying late on Friday afternoon, I would say. That's your off day, starts from here now. And yeah.
Norman's laughing because he knows I'm just going to go home and work, and then I'll work all weekend. But my job is evangelism, right? I do fundraising and evangelism. I hold the vision and the mission, and that's, it's just a leadership thing. This is part of that. And I got so much work, but thank you for being so considerate of my time. But really, it was a pleasure, and it was not a big deal to stay late if people were interested.
You were quite natural. That is why I think that reflected during my conversation. I would say.
I'm sorry?
I said you were quite natural. That is why it reflected the same with me as well. I think your energy was very positive for me.
Yeah, well, I've been a lover of technology forever. My first love was computers, right? And that was 45 years ago. And think about how computers have impacted my life, and my love of technology has. I'm still using them here. I actually, everything they set up, I understand it. And I do a lot of debugging of my own computers coming out of that world. But this is a technology that has, I think you're going to see that it'll have more impact on the world. The entire market for things attached to the internet is $3.5 trillion per year. Healthcare is $10.5 trillion per year. And that's only considering how little we can do to improve health. Think about that. Medicine is so limited, but it's about to do this. This is maybe a $30 trillion industry one day. Computers will be nothing. That's how much value this could give to human beings. I only loved computers because it would make our lives easier. They would connect us like we're connected here today, right? You know, that's why I fell in love with computers, because I just saw a future of, you know, easier stuff that allows us to just be more creative and do the things that we love instead of all the rote activities. Yeah, you know, formatting documents, adding numbers, building, you know, robots could build things, right? Instead of us having to build everything. You know, the technology comes with some policy issues, you know, right? I mean, we need to find a way where technology really benefits everybody in the world. Capitalism may not necessarily be the most natural system to bring it in the best way. You know, we're going to have to think about one day, you know, instead of saying that we need less people so those people are now disposable, we need to be able to say we need less people because we can take care of them without them having to really work. Instead, what they can do is pursue a good life. That survival is assured, and they don't have to, you know, build that into their family planning, for instance. They don't have to have 10 kids because they have to plow a field. No, the robots will do it for them. Maybe they have 2 kids, replace themselves in the world, and maybe the money just is there to support them, along with some discretionary money that gives an opportunity for entrepreneurs to lift their life above that, right? And you know, that's another part of things that excite me is, you know, my background is in economics, as you mentioned to everybody, and it's thinking about there's got to be a model by which, you know, we can make a better world for as many people as possible, right? Certainly computers have helped that. Certainly gene and cell therapy will help that. But also part of it is more than just a technology that brings it on. It's an attitude and leadership and vision for an economic model that has the widest benefits and doesn't hurt anybody, right? Like a doctor, do no harm, right? And I think that it's possible. I always think it's just ironic to me that there are poor people in the United States. I'm like, how's that even possible that there are people that don't know whether they have enough food to eat in the United States, the richest country on earth?
Richest country, exactly.
Yeah. Well, I think that, you know, within the United States, you can answer that question, but what you learn from that, you might be able to spread to the whole world, and you might be able to improve everyone's life. The United States, I don't want to sound like some sort of radical here, but the United States could be not just the most powerful country on earth and the richest country on earth, it could be the best neighbor on earth. It could be contributing to the neighborhood of nations, right? By bringing things out and not always thinking just about how do we make more money, right? How do we make somebody richer? Thinking that that'll somehow trickle down to the benefit of everybody else. Right? Anyway, there's so many problems on Earth to solve. I'm full up with gene and cell therapy stuff, and that's exciting to me, and I'm so happy to be part of this. I feel like the luckiest guy on Earth, and I'm hoping that we're going to see I'm hoping that we're going to see
I should say other way around.
What's that?
I should say other way around. We should be, you know, saying this to you, that we are lucky to have you.
Oh, well, thank you. Well, I hope I'll do something that will be, you know, will be seen as being good, you know, and that touches us here and not just, you know, money-wise or whatever. You know, that would be an incredible dream of mine. But, you know, to have been right place, right time, you know, and have the right background to do what I'm doing, and just have had the experience of being in all these technological revolutions, so that I was able to spot an opportunity here and to build this amazing company and to work with all these amazing people. Because, you know, Well, you're one of them now. You're part of the revolution. You're part of the AGT family because you understand our journey now, and so there's a little bit of a connection between us, and I think that's the way that all of these revolutions come on. But I look around my company, and this is not just a job. This is something that gives us fulfillment.
Passionate.
Yeah, we get something back besides the money, right? That if we cure HIV, that feels good. If you're a empathetic human being, what they call it helper's high. I saw this the other day on a show I was watching, I think on Netflix or something. But people don't realize, because we sometimes hold up greedy people as being a success, right? Rich people as successes, right? A lot of people don't realize that Mother Teresa felt really good. She felt better than Donald Trump every day. And yet she had nothing except for the fulfillment of helping others. That's how powerful that is. We still need to live in the world and eat, we need shelter over our heads and all this stuff. But to be able to have helper's high, like if we can solve HIV and get that good feeling, and we got paid for it, like people didn't have to give up their entire life. I mean, I took a huge risk, and I hope one day in retrospect people will appreciate that, yeah, Jeff and his wife put it all on the line to start this thing rolling. And Jeff went 24/7 and didn't get paid for 8 years, and and got no help on this until 2012, 2013, right? When all of a sudden that light attracted all this great rocket fuel for the rocket, right? And that they'll appreciate that level of risk and sacrifice and whatever. But to have survived that and to now be here and to have a line of sight on something that means so much to me is a blessing.
Thank you.
Yeah. All right. Well, thank you. Anyway,
Yeah,
it was great. I'm
thank
glad
you
it
so
worked
much.
out so well for both of us. And it was an easy conference to do. And you were a great moderator,
Thank you. Thank you so much.
Thank you.
Yeah, and in fact, you were quite natural, and I think keep doing what you really are doing right now. The passion and the energy and enthusiasm. Thank you
Thanks.
so much.
You're welcome.
Have a
Thank
good
you.
weekend now. Have a
Okay.
good weekend now.
Yeah, you too.
Yeah.
Okay.
Yeah,
Bye-bye.
bye.